Welcome to another session on food and its components. Today we are going to learn about test of the food components. We are going to talk about balanced diet, importance of food labels and malnutrition. So, we already know that there are different kinds of food components which include carbohydrates, fats, proteins, right? All these three kind of all these three kinds of resources or these three kinds of food components are required by our body in major amount. Now, a major kind of a carbohydrate which we consume on day to day is starch, as learnt earlier, right? So when we talk about starch, there is a method how we can identify starch. So to test for starch, what do we usually take? We take a slice of potato or bread. Now, any starchy food can be taken though, but usually potato and breads have high amount of starch. So we take a slice of the potato or bread, or even you can say the food material which you want to test for, right? You are going to mash this with a dropper and you're going to add a few drops of iodine solution to this. So iodine solution is in nature a brownish colored liquid, right? So a brown color liquid is what, or a brown color chemical is what a iodine solution is, right? In order to enhance this, what do? We add a few drops of iodine solution to your potato, and when we add a few drops of iodine solution to the potato, we see the color change happening. What happens? This brown color solution changes to blue black if iodine is present, if starch is present, right? Now if you see the diagram here, the image here, it says that you take a potato, you cut the potato or peel through a section of potato, and when you peel through the section of potato, the starch is basically exposed, right? Now if you add few drops of iodine, you can see here that the color has changed from brown to blue black. So blue black color will indicate the presence of starch in your food material. Okay. Now, similarly for proteins, there is another test for proteins, and this test is also known as the Biuret test, right? Yeah, Biuret test. Now, what do we usually do in this Biuret test? We usually take drops of milk. Why we have taken milk? Because we have learned that milk is a source of protein. It has amount of proteins and also carbohydrates, right? Or you can take egg white, and again any other food material you want to do the testing on, you can take that, right? Take that food material, and in that food material, now if that is a solid food material, what you will do, you will add a small amount of water and break it or mash it into pieces, and then you are going to create a solution out of it, right? And if it's like milk is already a liquid, egg white is already a liquid, right? So you can take them in the same form. You can add a few drops of copper sulfate solution, which is CuSO4, right? And then after adding few drops, you can add a few drops of sodium hydroxide, right? Now together, copper sulfate solution and sodium hydroxide together is known as the Biuret reagent. So either you add them separately, or you together in a combination, they are named by the name of Biuret reagent. Now when you add them, what you will see is that Biuret reagent, or basically copper sulfate and few drops of sodium hydroxide, is blue in copper sulfate is blue in color, right? So if this blue color changes to purple, right, or we also say as lilac color, so faintly purple or dark purple, then we say that the test, or basically the compound, or basically the food component contained proteins, right? And if not, if it just remains blue, like if you see here, it's a negative test, means proteins are absent, right? Now if I do a similar test on any other compound, I can do it by this method to test for proteins. So a violet color, a blue color, a lilac color. So the deeper the color, Like if it's violet, it has more protein. If it's lilac, it has less protein. If it's purple, it has a medium amount of protein. So that is how we test for proteins. Okay. Now, there's also a test for ethanol, and this is known as, basically, there's also a test for fats, and this is known as the ethanol emulsion test. What is an ethanol emulsion test? So, ethanol is a type of alcohol, right? Now, what do we do? Again, you take a small amount of food sample. So, usually, if I have to test for, let's say, if I have to test for a fat, I am going to take oil. Maybe I can take an oil, right? I can take any kind of oil, maybe an animal source of, maybe I can also take milk here to test for ethanol emulsion test, right? Now I'll take the food sample, right? So oil or maybe any kind of ghee or any kind of fat is usually liquid form, right? So like I told you, so you will take liquid form of it, right? So take the small amount of the food sample in a test tube. Add equal volume of ethanol to the sample. So you'll take the food sample. Suppose you took one, you took 4 drops of oil. Okay. Add 4 drops of, so like equal amount of ethanol, equal volume of ethanol, 4 drops of ethanol. Okay. And then you will vigorously shake the test tubes for a few minutes. So just try and mix them. Now, when you will add this, you will see then when you keep the test tube for a few minutes for a while, the milky emulsion forms upon standing, right? So you see that a milky, basically cloudiness. So if this is your test tube, so a cloudy kind of appearance you would see on the top of it. That indicates that fats are present. So it is also known as the ethanol emulsion test.